Bacteria evolved before humans and evolved more quickly than humans. That's one of the take-home messages. Bacteria are present in our body and everywhere in our environment. Both bacteria and humans sense each other, and this interaction leads to inflammation, disease, immunity, and health, and we're going to talk about that. And humans have diversity in their response to bacteria, which might contribute to disease outcome. So for those of you who don't know, what are bacteria? And bacteria is a plural. If it was a single bacteria, it'd be bacterium. And they're the first free life form. So when we stopped being molecules on Earth, when it stopped being just RNA and DNA and molecules, this was the first free life form that had a membrane or had a barrier between it and the rest of the world. They're just a single cell. They have no nucleus. In contrast, human cells have a nucleus or a membrane that contains the DNA and does all those functions. There's a cell envelope for protection, and we'll try to explain that. And many bacteria, though not all, are motile, and they have environmental sensing. So they're actually much more complex because they're much older, and they're actually much more evolved than we are in some ways. So what's the evolution of bacterial virulence and specialization? So all this specialization of bacteria, it started out perhaps as one bacteria, and then they began to specialize. And so they specialized. Many of them produced antibiotics so that they could compete with other bacteria. In fact, all the antibiotics that revolutionized medicine in the 20th century and changed all of our lives all came from microorganisms and many from bacteria. They have specialized cell walls. They're able to transfer genetic material and lose genetic material, which allows them to adapt pretty quickly. They can secrete enzymes, or what we call toxins, which are proteins with specific biochemical activities that can do things to other cells. They have specialized sensing, specialized metabolism, and then they adapted to animals. In fact, animals are like a giant food source for bacteria. And, bacteria are part of our body. As Elhanan may have told you in his lecture, I think, sometime in the last few summers, there are about 10 times as many bacterial cells as human because they are small. And they're essential for normal development of our intestinal and mucosal surfaces and our immune system. And they evolved with animals. Animals provide food for bacteria in a more constant environment. If you think about if you're a bacteria and you're subjected to climate change, you're like a plant standing out there. Well, it could get cold, it could get hot. But when you found animals, they have a constant temperature. We're all 37 degrees. And so they could have more specialized adaptation. And there was food there, good food, and it was easy to get. So I even wrote a review called "Are Pathogenic Bacteria Just Looking for Food?" because I think that's the driving force for pathogenesis and the evolution of virulence for bacteria is to get food and to replicate. So because that's the driving force for life. Now, bacteria, in addition to helping us with our development, can also be pathogens. And they cause both acute and chronic diseases. And I don't want to take you through all of this, but they essentially cause disease in every part of your body. And all the diseases that you might have had, diarrheal diseases, food poisoning, pneumonias, bronchitis, systemic illness that can lead to death with bacteria in the bloodstream, all the wound infections in the skin, infections of the nervous system and of the urinary tract and your sexual organs. So they cause disease everywhere. And they're actually the most important for global mortality. If you look at this global mortality report, which is a little bit old, but it still holds, the percentage of all deaths and the number of deaths, the highest is respiratory infections, of which about half are bacteria in the world. Tuberculosis is caused by a bacteria. And diarrheal disease, about half of those are bacteria and about half of those are viral diseases. So these are what's causing death and also promoting our health. Now, bacteria can be dumped into three groups by staining them with dyes. And this is Hans Christian Gram developed the Gram stain. He was a Danish microbiologist in the 19th century, and he discovered, after the microscope was discovered, bacteria were discovered because you needed a microscope to see bacteria, and you could see them better if you stained them with various dyes. And they went into two classes, those that were blue and those that were red based on those dyes. And Paul Ehrlich, a German Jew who discovered tuberculosis, discovered that there were bacteria that he could stain, and that stain remained red in the presence of acid. So these different stains are because bacteria have different cell walls. And the cell walls are complex chemical structures that really protect these bacteria from noxious substances. And this is going to be part of what we sense as humans when we sense bacteria. And so these gram-positive bacteria make a lot of this sugar peptidoglycan on the surface, but only have one membrane. And the gram-negative bacteria had two membranes, including one of which had this lipopolysaccharide molecule, which is a glycolipid on its surface. Sorry if this is too chemical for some of the students in the audience. But it's made of fat. This is a molecule made of fat and sugar. Now, why did this evolve? Because it's a tight barrier. The gram-negative bacteria evolved. So bacteria probably started out with one membrane, and then they evolved a second membrane. And that's because they could protect themselves more from noxious substances. And this is a permeability barrier that excludes hydrophobic, and it has a negative charge, so it excludes positively charged molecules. And there are specialized molecules that control the flow of various soluble molecules through them. And they have pumps to pump things out, and these two phospholipid bilayers. Now, these primordial gram-negative bacteria also contribute to our life, in that we believe that most of our energy machines, the mitochondria, which have two membranes, evolved probably from intracellular parasites that then took hold and remained in eukaryotic cells, because they both have specialized two membranes, as does the chloroplast, the energy engine for plants. So this is sort of contributing to our eukaryotic life, both in terms of our development and also probably in terms of how we developed as eukaryotic cells and improved our energy acquisition. So what are bacterial variance factors? Why do bacteria, we're going to now talk about what makes a bacterium virulent, what allows them to cause disease. And one is that they have this barrier to host killing that helps them survive in a niche. So if they're driven for more food and they move from the interior of your intestine to invade your tissues or the cells of your colon or of your bowel, then they have to have barriers to survive the host killing factors. They have to be able to adhere to cells that allows them to colonize. They have to be able to invade cells, which is a specialized property of just going into a eukaryotic cell. And they have toxins, or these proteins that are enzymes that alter human cells through killing them or through some other mechanism that manipulates a host or a human cellular process. And then there are these nutritional factors that promote growth in a new niche or environment. So some pathogens have specialized metabolic pathways that allow them to eat food that they only get in a human setting. So virulence is an evolutionarily moving target because bacteria are evolving over years. We're evolving over, millennia or over 50,000 years or 10,000 years, but these bacteria are evolving over years. And humans evolve more slowly, and humans evolved an immune system to deal with this problem. So why aren't humans all dead? Why aren't we all dead? But these bacteria can evolve in leaps because they can acquire DNA through mobile genetic elements. And so there's most of the classical virulence factors that have been studied since the beginning of time were acquired in these evolutionary leaps. In other words, we didn't start out, bacteria don't just have their single chromosome, which they have, and then have that evolve for them to do it. They acquire DNA either through bacteriophages, which are like little viruses that can inject DNA, and most of the famous toxins that cause cholera or diphtheria that all of you are vaccinated with as children against diphtheria, a disease that causes a severe sore throat and cause childhood death. We're all vaccinated against diphtheria, and we're vaccinated against the diphtheria toxin that was acquired by just a normal bacteria when it got this toxin on a phage. And then there are other complex whole chromosomal regions which recombine between bacteria that allow them to get large chunks of DNA. And then there are plasmids, which are these self-replicating DNA elements which cause antibiotic resistance. So these host pathogen interactions then sort of start out as warfare, but they evolve toward a detente. And many of you are probably too young to even understand detente. I think I made this slide or had an artist do these drawings after the Cold War ended, which tells you how old I am. But detente was something that was part of after the Cold War. In other words, the perception 20 or 30 years ago was, bacteria produce a toxin and they kill you, and there's a warfare going on between the host cell and the bacteria, when actually it's much more subtle communication, more like an economic advantage. And that sort of tells you the recently evolved pathogens are more virulent. So if we take Shigella, which is a gram-negative pathogen that recently evolved, some estimates the most epidemic Shigella evolved maybe only 100 or 150 years ago by a normal E. coli acquiring one of these mobile genetic elements or a plasmid. And this plasmid allowed this organism that was just living in your colon and helping with normal life and living in symbiosis with you to then invade host cells and then to polymerize actin. And this image just shows how it can move through the cytoplasm of a cell. It breaks out of a vacuole that it enters into and then can invade another cell through these polymerizing actin things that cause it to interact with another cell that's in close proximity and invade and replicate. And the drive for this was probably the acquisition of food, or it had to go perhaps even starvation. And this caused huge outbreaks as part of World War II and World War I as part of war. And that's a human-specific pathogen that recently evolved. Whereas something like Salmonella, another gram-negative pathogen, evolved as an intracellular pathogen to stay within an intracellular vacuole. And this is an old pathogen that's been associated with animals for a long period of time, particularly what are called the non-typhoidal Salmonella. And so this ends, if we get Salmonella and we're healthy, we have 48 hours of diarrhea and then we get better. And that is a complex interplay. We get better without antibiotics. So somehow there's this huge complicated mechanism where the bacteria enter host cells and do all these complex things to the host cell, and then it's resolved. So that's really, when we've had pathogens or had them in our lives for a long period of time, it reaches sort of an equilibrium, or else one of the species would die out. So why were animals not wiped out by bacteria? And how can we control Salmonella and many of the other pathogens? And how did we even evolve as humans before we had antibiotics, right? Why aren't we all dead? Why are we still here? And that's because we evolved an immune system. And there's two types of immunity. Innate immunity, which is said to be not learned, and adaptive, which is a response to a specific stimulus. So if you have a very specific, if you're vaccinated with diphtheria toxin, then, or toxoid, as a child, you make very specific immune responses that protect you against that toxin and cause you not to have disease, even if you're infected with diphtheria. But these innate immune responses are non-learned. They're part of our basic makeup. So what's the disease? The disease from bacteria is actually protecting us from death. This is inflammation. And so how is this, what is this? Now we call this inflammation. So we sort of call disease inflammation. And Celsus, in the 1st century AD, defined using these Latin terms, since he was a Roman, what are the signs of that? And there's calor, or heat, rubor, redness, tumor, or swelling, and dolor, or pain. And these are the signs of inflammation that we used to be taught as medical students, but probably aren't anymore, these old Latin words. And how did this inflammation occur? Well, there's a big, and this is what innate immunity is. And we understand now innate immunity in a much more complex molecular way today. But what it really is when you breach this barrier, and we have all these barriers to keep the bacteria at bay, and to keep the microorganisms at bay, and to keep particles at bay. We have our skin, we have all our mucosal surfaces that line our mouth and our intestinal tract, and all those cells are creating barriers and mucus on the outside, and so these bacteria can't get across. But when you breach that barrier with trauma, then there's a lot of reaction. Bacteria get in there, and there's a huge reaction with fluid rushing in, host cells coming in that can eat these bacteria and kill them, and a whole bunch of different molecules to deal with this problem. So that, before we had antibiotics, we would have an infection in our foot, and we'd keep it out of our bloodstream with inflammation. Now, Charles Janeway, who's now deceased, but was a famous professor of immunobiology at Yale, and sort of coined the term innate immune system, because the study of the immune system for a long time had been driven by transplantation, and the idea of transplantation and self versus non-self, transplanting something from a human to another human, or a mouse to another mouse. But his idea was that the immune system does not discriminate self from non-self, but rather could discriminate infectious non-self from non-infectious self. In other words, that it could discriminate infection, that was the evolution of the immune system. Not about transplantation, but it was actually about bacteria. And there's a lot of different innate immune mechanisms. As I said, there's mucosal barriers, there's cells that eat other microorganisms and kill them. There's a variety of different proteins that recruit different cells that are called cytokines and chemokines. Then there's antimicrobial peptides and detection of pathogen-associated molecular signatures. Now, antimicrobial peptides are among the oldest of immune systems. And I think I'll show you some slides of them, but they're made by bacteria. They also make up the entire immune system of most insects. Now, these cell surface receptors and pathways that sense microbial products to activate inflammation. And you don't have to understand this slide just to say that there are a variety of different cell surface receptors that are critical. And these were discovered in the last 15 or 20 years. The Nobel Prize was given for them in the last couple of years. They were originally discovered through work in insects by Jules Hoffman, and then Bruce Beutler also won it for the LPS receptor that's on the surface that recognizes, and they typically recognize things on the surface in those cell walls of bacteria. They also recognize various viral products. But there are also cytoplasmic receptors are also critical and can indicate more severe response. So you have these receptors on the surface telling you bacteria are close, but if you get some bacterial products inside your cell, in the cytoplasm, well, that's more serious business. And in fact, it's recently been shown that there's an LPS receptor just in the last couple of years that it determines whether you live or die if LPS gets into the cytoplasm when you're in sepsis. And so intracellular pathogens that go to different locations. As I told you before, salmonella end up going into a vacuole, and so does tuberculosis. But there are other ones that escape out into the cytosol, such as Shigella, and they engender more severe responses or more acute responses. So what is this complex conversation between bacteria and humans that results in inflammation? And so how is this conversation between the bacteria and the host cells occur? And so bacteria first have to sense they're in a specific mammalian environment. And this is a principle of bacterial pathogenesis that bacteria are not a simple organism. They sense very specifically where they are through cell surface receptors, and then only induce virulence factors when they're in the right place in the right spot. And they respond to this environment to maximize their survival and replication in this new niche. And host cells then sense bacteria and elicit inflammation. The process resolves or not, but the pathogen benefits by that replication and evolution. So that's sort of what we hope to illustrate. And so here are these principles that I like to say of bacterial pathogenesis. An organism first has to sense it's in host tissues. It has to remodel its surface, or this cell wall, has to then be remodeled so it's not killed, and then it has to modify host processes. And it can modify them by secreting a single protein, or as I'll show you, these specialized interspecies protein transport systems that evolved to transport dozens of proteins into a host cell, such as many gram-negative pathogens do. Now, the first experiment that showed coordinate regulation of virulence by the environment was done by Pappenheimer, who was a professor first at NYU and then at Harvard. And this was very important during World War II, because soldiers, diphtheria, I told you, causes a severe sore throat, but it also causes wound infections, because the bacterium that causes diphtheria is in the dirt. And during World War II, wounded soldiers were dying of diphtheria wound infections. And Pappenheimer, the government called on Pappenheimer, who was studying diphtheria toxin, to help them to make more diphtheria toxin so they could make vaccine, because it was already known you could take diphtheria toxin and treat it with formaldehyde, and it was inactivated and you could get immunity if you were vaccinated. And Pappenheimer discovered that when he grew the bacteria in a low iron media, they made a lot more toxin. And now we know the whole molecular basis of it. But you could show you that sometimes understanding this kind of stuff has practical importance, because it saved many soldiers' lives, because we couldn't get the bacteria when it was grown in culture to make very much diphtheria toxin until we figured out how to grow it in low iron. Now, these hosts, just like I told you that the host cells sense the cell wall, bacteria sense the host. And they do sense a specialized nutritional environment, and sometimes they sense a specialized temperature environment, but they also sense these antimicrobial peptides. They're an important signal for virulence. And these antimicrobial peptides, as I told you, are widely distributed in animals and plants. They're inducible in mammals. As I said, they're among the most ancient host defense factors. And most have cationic properties. So most of them are kind of like a detergent, a very sophisticated detergent. If you learned in high school biology what detergents are, right? They solubilize, they have an interface between all that grease and the non-grease, right? So they're amphipathic. They have a charged surface and a lipid soluble. And these are like that. They have a positively charged surface, and they target all these negative charges on the membranes of these bacteria because they are positively charged, the bacteria are negatively charged, and then the hydrophobic component or the lipid component inserts into that lipid membrane and disrupts it and just blows holes in them. And they kill by destroying the membrane. And they have a lot of different structures. The most ancient one is actually still used as an antibiotic that's produced by gram-positive bacteria to kill gram-negative bacteria. And then there are alpha helical peptides, which is another structure. These make up most of the immune system of insects. And mammals evolved these beta sheet structures because bacteria produce proteases that will degrade these. And bacteria have sensors that measure the environment and change gene expression. They have antimicrobial peptide sensors that then sense this environment and change the chemistry of the cell through phosphate transfer, which is one of the ways that all organisms communicate across membranes is by phosphorylation. And then when they sense these antimicrobial peptides, they put their shields up. When I first made a discovery about this, I was once interviewed by the BBC, and they asked me to explain it to people. You know, we discovered that the LPS molecule could be remodeled in response to this. And I said, well, it's like Star Trek. And, you know, the bacteria are just putting their shields up because they want to protect themselves against these antimicrobial peptides and all the noxious substances. And so they remodel every component of this membrane, the protein components, and many other components, which is too detailed to go into. They also, when they go into a host cell, they repress the synthesis of these organelles recognized by the innate immune system. These flagellar organelles that help with their motility are like, that allow them to swim. This is a circulating organelle. And these interspecies protein transport systems that I alluded to, or the translocon. And then they change their LPS surface basically by making it more hydrophobic. So this glycolipid has all these fats which sit into the membrane, and it has these phosphate molecules that give it the negative charge. But if you chemically modify all these phosphates and all these sugars, you reduce the charge. So they're basically becoming less charged as a way of putting up their shields. And then when they alter host processes, they do that by directly injecting toxins into host cells. And there are at least 10 different ways that bacteria deliver proteins to host cells. But one of the most evolved is that they evolved something from flagella that's like a needle or an injectosome that is this complex organelle which docks with the mammalian membrane and allows them to inject up to 20 proteins through this apparatus to alter every aspect of the cell. And they alter the innate immune response, they alter the cytoskeleton, which makes up how cells move and their structure, sort of the bones of the cell. And they can cause cell death or cell cycle arrest. And what does this look like? So when Salmonella enters these cells, they dock with this injectosome and inject a number of proteins that cause the membrane to open up and engulf the bacteria. So they trick the host cell by manipulating these specific molecules for the scientists in the audience, Rho GTPases, to allow the bacteria to be taken up. And that's just one of many, many examples. So what determines, one of the important questions I think that remains as we start to have lots of advances, such as what's going on in genome sciences today, as we start to have lots of advances in human genetics, is what determines the differential outcome of infection. And this has been a question for a long time. And all these different ligands that are sensed by this innate immune system can be quite different. They're different in different organisms. They're different and bacteria have the ability to modify them, so they're not recognized as much. And then there's all this different innate immune diversity because organisms are so completely different. They all have evolved different mechanisms. But we still, for humans, we know from many different outbreaks that lots of people are infected, and some people don't get any disease. And some people have acute disease or chronic disease, and some people die and some people don't. And so why is that? And is that going to be a powerful tool for us to understand disease today? And, you know, pathogens have driven evolution of the human genome. There's a lot of evidence that the genes that are under natural selection in the human genome are most associated with immune responses, both immune responses and starvation, which are also probably linked to, have been shown to be linked to immune responses. And bacteria are sort of unique probes about mammalian processes because we've co-evolved with bacteria. And they can reveal important points of selection pressure that may influence other types of human phenotypic diversity in addition to infectious disease outcomes, such as chronic diseases. Now, what's the evidence for this? Now, genetics does strongly increase the risk of death from infectious disease. If you look at this older paper from the New England Journal of Medicine that are twins, some separated at birth and adopted, you can see that the relative risk, if the biological parent is dead before 50 from infection, is almost 6 times. Whereas with cancer, it's the opposite. The relative risk if the adoptive parent is dead before 50, suggesting that this, at least in this study, was more environmental. Obviously, there is some cancer that's genetic, and there are genetic components to cancer, but at least in this sort of crude cut, this appears to be more inherited than cancer. And we've known for a long time that common alleles confer resistance to infectious disease. Many years ago, it was shown in the 50s that hemoglobinopathies or anemias, you may have heard of sickle cell anemia, and other thalassemias or other anemias or abnormalities of our red blood cells that carry our oxygen. There's good evidence that they evolved to protect us from malaria. And this was in atolls where there was these isolated islands where at high altitude there's no malaria, at low altitude there is malaria, and the hemoglobinopathy frequency tracks exactly with malaria. And then we've known for a long time that a specific chemokine receptor or an innate immune receptor, the CCR5 receptor, if you have a mutation in that, it protects you from HIV. And recently, the evolution of this is probably related to a bacterial toxin, a leukotoxin produced by Staph aureus, which causes skin infections and other infections because it's the receptor for that toxin. So probably the selection pressure for this was not HIV, which moved just in this recent last century from animals to humans, but it's probably bacterial toxins. And we know that for many diseases, there's a difference in death rate. And one of the greatest examples of this, in the 19th century, yellow fever swept the South. And there was a really high death rate in New Orleans and Memphis from yellow fever, which is a viral disease that's most common in West Africa, actually, that causes death of your liver. And it was actually believed at that time that the disease could have been transmitted from African Americans. And there was persecution of them. And this man, John Erskine, in Memphis, actually did an epidemiological treatise that proved that it was not, they didn't know it was transmitted from mosquitoes. That was only discovered by Walter Reed about 20 or 30 years later. But he was able to show that it really wasn't associated directly with what appeared to be transmission from African Americans, but they were actually protected from it. So their death rate was very low, whereas the death rate for Caucasians was 70%. Probably this was because African Americans had been enslaved and shipped from West Africa where they'd been exposed to yellow fever, and there'd already been selection pressure on their genome, allowing them to survive a yellow fever outbreak in the South, whereas the Caucasians may never have seen it before because they all came from Northern Europe. Now, we've been doing, now it's common in the new era of genomics and of having all these genome sequences to do what are called genome-wide association studies, which are to take natural populations and to try to see if there are associations of particular genetic variation, which we can measure, with some specific outcome. And there have been some of these done for infectious diseases. But the problem with that, there are some valuable things that come out of that, but it's somewhat limited. And so one approach we've taken, and others have taken for cancer chemotherapy and pharmacogenetics a little bit, is to do cellular GWAS, which is a complementary approach, which is to use cells from people to do these kinds of association studies. And it reduces the environmental variation due to differences in dose, medical treatment, provides clues to mechanism, allows for experimental follow-up, and not restricted to diseases that have large, currently accessible patient populations. And it's somewhat accessible to small labs. So if you want to study how humans evolved virulence from plague, or if you thought you can't go out and find a large population of people with plague today. And so that this allows you to study that. And so we've done this by studying this variation, by measuring phenotypic variation in cells from hundreds of different individuals, and identify these genetic differences, and assess the relevance to human health and disease. Now, what we have done with this is use these cells that are EBV transformed B cells that are part of a large project of a catalog of common human genetic diversity where all the sequences of these cells live. But what's evolving now is with stem cell research and the ability to differentiate stem cells is to have hundreds of non-transformed cells. And recently, Stappenbeck's lab at WashU in St. Louis has differentiated over 100 epithelial cell culture lines that appear to have normal differentiation and make mucin and stuff. This is a huge breakthrough from hundreds of different individuals now. And so there may come a time in the future where we all have our cells, or where there are many, many cells that are normal cells that exist in culture, that we can actually begin to characterize these types of interactions, and not just some B cells that have been infected with a virus. But it actually worked for us to do this sort of phenotyping of human cells with bacteria and various products, and it's allowed us to discover some human variability in this process. And for instance, one of the complex problems with complex diseases like this and these common genetic variability is trying to determine something that's due to many, many different genetic variation points. That's been a real challenge with, say, diabetes or with many of these sorts of complex human diseases. But we were able to find the heritability of, and over 4 SNPs explain the heritability of one important innate immune response, at least in these 500 or so individuals. And they explained about half of that variability. So we're able to determine that. So what does that mean? And I think if you put this into some historical context, you know, we've only been around about 6 million years ago as humans. And about 10,000 years ago, there was the rise of agriculture and animal domestication. And with that, there were the beginning of cities, which occurred about 5,000 years ago. And at the time that humans came together in large groups is probably when infectious diseases became a big problem, because it was easier to transmit them, because infectious diseases are transmitted from people to people, animals to people, and in large groups. That's where you get big outbreaks. And so this is the period where there's been selection pressure on those people who acquired agriculture earlier, not hunter-gatherers. And so we have data to suggest the hypothesis that humans sort of evolved as herd animals, and that are becoming like herd animals. And one of the alleles that we found, a null mutation in CARD8, is actually animals that live in large herds or colonies are likely to have lost CARD8. So that's one particular regulatory innate immune gene. And we're not going to see loss of whole innate immunity, or if we do see major effects on the immune system, then you usually have children. We do see that sometimes, and then children die at a young age. But in these common alleles from normal humans, what we're seeing are that like the animals that live in large herds, we're beginning to degrade and lose CARD8. And this CARD8 allele we showed with Mark Warfel is associated with higher risk of a bigger inflammatory response when you're infected with bacteria or present to the hospital with what looks like acute blood infection. And also other investigators have shown this allele is associated with an inflammatory disease, increased risk of rheumatoid arthritis, suggesting that maybe the price that you're paying for protecting yourself from bacteria that was selected at the time of civilization occurring is that we're going to have more autoimmune disease as we get older. And that doesn't explain all the increase in autoimmune disease over the last 40 or 50 years, but could be part of it. And, you know, hunter-gatherer populations have a lower frequency of these alleles. These Pima Indiids and the San of Navia have a much lower frequency than the CARD8 allele. And then we also showed one of these other alleles has a role, again, in this inflammatory response, but also has a role in nutrition. So that other allele that we found that's important for the inflammatory response is in a gene called APIP. It's involved in what's called methionine salvage, which is an amino acid. So when you're starved, you have to make methionine. If you need to make methionine, then this pathway cuts in. But since many of us maybe don't have to worry so much about starvation or not, many of us, particularly more of us who acquired agriculture earlier, have lost or have much decreased ability to do methionine salvage. And that results in a more robust inflammatory response through this intermediary. I don't have time to tell you all about it, but just the principle. And so there might be this nutritional switch for a host response in infection. So early in infection, you're going to sacrifice cells because this response kills cells. We kill the bacteria by killing our cells with the caspase-1 response. And when we're nutritionally replete, we can afford to kill ourselves to get rid of bacteria. But when we're starved, we probably can't afford to kill ourselves, and so we need to get rid of that response. But again, that allele, I know I'm going over this quickly because I want to leave time for questions, but this allele was also associated with decreased mortality for this sepsis-like syndrome, or bacteria in the bloodstream, of twofold relative risk, which is pretty significant. And it was associated with decreased culture positivity in those patients. So this suggests that we may have an optimal inflammatory response in humans. We're not going to get rid of, this inflammation response. Mostly what we study as scientists, we study a lot of mice that are knocked out for specific pathways. And so if you knock out the whole pathway, but humans are not going to evolve to knock out the pathway. They're just going to fine-tune the pathway. And so they're going to get alleles that cause loss of function that will increase these responses, which will protect them from bacteria. There'll be selection for that, but there may be increased susceptibility for inflammatory diseases. And it's many of these screens. We've done lots of screens now, and this seems to be a general principle that we're finding. So, as I said, this natural selection, agriculture, animal domestication, denser populations, more infection, crowd diseases, and then polygenic selection for decreased function or expression of inflammatory responses. And we can see this in the history of human genomics, where agriculture developed earlier in Europe and Asia, and these alleles, like the APIP allele, is found in higher frequencies in Eurasia. And it's one of the alleles that's most associated with agriculture sooner. So I'd like to conclude and say bacteria also cause chronic diseases, and there's much interest that they may help understand many other diseases of modern humans, from atherosclerosis to diabetes, because increasingly all these chronic diseases have inflammation as a common point. And many people believe that bacteria are contributing to these diseases in many ways, and neurological diseases. And we already know that in my lifetime, when I went to medical school, I was told that ulcers were caused by acid. But of course, very early in my career, it was discovered that it was caused by a bacteria that commonly lives in the stomach of most humans, except those of us who are in the developed world. And the same thing is probably true for many other diseases. And so I hope I've told you tonight that bacteria are interesting and that studying them is worthwhile. And I covered a lot of material, and I'd be happy to answer any questions you might have.